Hello everyone. Sometimes I saw interesting fungus growing on plant or other stuff. I'm thinking if I can isolate and culture them. Well, there are several different ways to isolate fungus, and in this video I'm going to show you one of those methods and also how to clone a mushroom that I bought from the market. First, we have to prepare something. We need an alcohol lamp, some clean container. Here are some glass petri dish, some sterilized tweezer or clean or sterilized tweezer. Or for some sample, you might need a surgical blade. And I'm using the number 11 one. Some more clean container. You can get this kind of container or the small one. Like you need 3 of them. Yeah. Some sterilized water. Some marker pen. Gross medium agar plate. Here I have 2 of them. And some sterilized paper towel. Like kitchen paper towel. Here are the house bleach. It's diluted house bleach. It's 10 times dilution for the surface sterilization. I'm going to use 2 types of specimen to demonstrate. One specimens are those leaves. You can see those black spots on the leaves. They are actually some kind of fungal infection. Another specimen is this potato. And you can see here there are some clearly like some fungus are growing here. Now let me set up first. I'm going to put the sterilized water into these 3 containers and bleach into another one. Or some people will suggest using a sterilized container, but most of the time I just use clean. I just wipe them with ethanol. And here, well, my flame is unstable, so let me go get a windshield. Now the leaves, and here I realize that I don't actually need to use the surgical blade on the leaves, but I open it anyway because, well, I'm going to use it on the potato though. So for the leaves, I can just use a scissor to cut the black area. I cut the leaf into like further into like smaller pieces. The important thing is that here I'm cutting the area with the black and also like part of the surrounding tissue. Like I'm not actually taking like all the black area, but also the surrounding tissue. We call it like it's some kind of a transition between the disease part, the infected part, to the healthy part. You know, we want that area in the middle. And you don't want the pieces to be like too big. Like I think it's somewhere around like 4 milliliter square, so it's like 2 times 2. Or like a small like a half gram of wheat or rice, I guess. Now what I'm going to do next is to surface sterilize those pieces. The first step is to sterilize them with alcohol, 75% alcohol. Simply just spray the pieces with the alcohol and let them soak for anywhere between 2 to 10 seconds. I think it's enough. Here I'm doing it for like 5 seconds. And then transfer the pieces into diluted bleach, which is usually like 10 times dilution, and let them soak for anywhere from 4 to 5 minutes. After this sterilization, the next step is to use water to wash off the remaining bleach and alcohol by transfer those pieces into the clean sterilized water, and that then soak for like 2 minutes, and this process will be repeated like 3 times. So that's why here I have this 3 container here. After I'm done with all the washing, I'll use a sterilized tweezer to pick all the pieces and put them on the sterilized paper towel to dry the water. After drying, I can put those pieces onto agar plate, and I usually use potato dextrose agar and water agar. Now done with the leaf, let's move on to the potato. So first what I'm going to do is the cutting the potato. And the principle is the same as the leaf, that you want the small pieces of the area between the healthy tissue and the infected tissue. So here the part is the part you can see the area that the color has become darker from the potato, like white potato color onto a darker color. That would be the area. Also, yeah, cut them into small square chunks. Yeah, small chunks. You don't want the chunks to be like too big. The sterilization process is the same as the leaf. The only differences being the potato will soak more time in the bleach, from 5 to 6 minutes. The rest of the process will be the same. It will take few days for the fungus to grow on the agar plate, so I usually seal the plate with a double layer parafilm. Now let's move on to the mushroom cloning, and this is the mushroom I got from the market. It's called the Black Beauty, which is a cultivation variant of the king oyster mushroom. The cloning process is pretty simple. I just take part of the mushroom and then spray it with alcohol to clean the outside. And then what I'm going to do next is to tear the mushroom apart, because we want the inside of the mushroom, which is theoretically clean, uncontaminated by the outside. And I use this sterilized tweezer to take a little bit of tissue and transfer it onto agar plate. And we're done. After a few days, you might see some mycelium growing out of the sample, and it's time to subculture them onto a new agar plate. I usually use a sterilized toothpick to do this. Use a toothpick and to pick up a tiny bit of mycelium from the edge of the colony, then transfer it onto a new agar plate. And we're done. Now let's look at the result of our isolation. First one is from the leaves. You can see here the colony from the leaf tissue. They both have their distinct color. And the second one from the potato. White mycelium grows pretty well. And this one is the mushroom. Still white mycelium. The colony looks pretty. I often use this technique to isolate fungus from all types of specimen. Here are some of the examples. You can see all those colonies, they look interesting, unique, and beautiful. Now you can use this technique to find interesting fungus yourself. I'll see you next time. Bye bye.